So first, I'm gonna draw out the membrane, the plasma membrane. This is the plasma membrane, and what we have attached is the G protein coupled receptor. So this is the G protein coupled receptor. I'm gonna label that GPCR. And then there is some ligand over here. Right, there's some ligand over here. And in this G protein coupled receptor scheme, we also have an effector over here. So I'm gonna draw out an effector. And let me label the outside as extracellular. And here we have intracellular. Okay, now what do we think we are missing? So at this initial resting stage, we also have a G alpha subunit and a G beta and gamma subunit. And they are sitting inside the cell, bound to the inner membrane. Okay. So we have a G alpha subunit, and in a resting state, so it's lipid anchored to the inner membrane, and this one is G alpha. And in its resting state, it is also bound to GDP. It's bound to GDP. All right, and then we also have G beta and gamma. G beta and gamma are also bound. They are bound to the G alpha subunit at first, at the resting state, but they also lipid anchored. So G beta and gamma is going to be here, over here. So we have G beta and G gamma. So I'm just going to write them as G beta and gamma over here. Okay, so this is the initial state. And now, so initially the three subunits, alpha, beta, and gamma, they're joined together. Okay, G protein coupled receptor is sitting empty in the cell membrane. And the alpha subunit is tightly bound to a molecule of GDP. And because it is holding a GDP, the system is off. And then the next step, a ligand binds. So the ligand is shown in blue over here. So this ligand could be something like a hormone, neurotransmitter, or older molecule. It binds to the outside of the GPCR. So let me show that binding over here. So we have this ligand here, and it's gonna bind like that. Okay. And then after binding, what do you think happens? Yes, right. There is going to be a conformational change. So binding leads to a conformational change, okay? And it just means that the GPCR is gonna undergo a changing its three-dimensional shape. And this changing shape is gonna activate the receptor on the inside of the cell. So now it's activating the receptor on the inside of the cell. Okay, so this is activated. GPCR inside, the receptor inside is activated. And now the activated GPCR, it binds to the nearby G protein. Okay, so it's sort of like, it's gonna act something like a crowbar, and it's going to force the alpha subunit to drop its empty GDP. So alpha subunit is drop its empty GDP, and because the cell cytoplasm is full of GTP, a fresh GTP molecule immediately swoops in and binds to the empty alpha subunit, and now the system is on. So let me show what happened. So we have ligand activating the GPCR, and now GDP is swapped for GTP. So this conformational change makes this GPCR closer to those resting subunits over here, and the ligand binds. So what happens now? Well, those subunits over here, they are going to move. Then they don't move, but rather the GPCR, the conformational change, makes them closer together. Okay, so now they're like close like that, like they're bound like this. And then when it happens, the GPCR also displaces the GDP, so GDP is now being displaced, and because the cytoplasm is full of GTP, so this GDP is, once it's gone, it's immediately being replaced by a GTP. Okay, so now this is a GTP. Okay, very straightforward. And then, what do we think happens next? So next is actually the association. So the binding of GTP changes the shape of the alpha subunit, causing it to lose its grip on the other subunit. So it's gonna detaches. It's gonna detach from the other subunit. It's also gonna detach from the GPCR itself. Okay. So the G protein is gonna split into two active pieces. We are gonna have so it's gonna split into the alpha GTP subunit. The G protein is gonna be split into two things. One is the alpha GTP subunit, and the other is the beta gamma dimer, and they are still gonna be glued together. Okay. And so what happens is you have this G beta gamma subunit, and it's actually going to slide along the lipid anchor, and then it's going to, its target is just going to be the GPCR. Okay, so that is that. But then the G alpha GTP is going to detach. Okay, so this one is going to detach, and yeah, so it's going to detach. It's going to come over. It's going to go to the effector here. Okay, it's going to come and bind to the effector, just like that. Okay. So it binds to the effector, and this G beta gamma is still, okay, it is still gonna be, it's gonna keep binding to the GPCR over here. So okay, so it's gonna bind to the GPCR like that. Okay, after dissociation, and we can see this G alpha GTP is now bind to the effector. So what do you think it does? Well, obviously it's going to activate the effector. So it's going to activate the effector, and some common effectors, like it could be something like adenylate cyclase. So examples could be adenylate cyclase, adenylate cyclase. And if alpha subunit turn this on, it produces a massive amount of second messenger called CAMP, which goes on to activate other kinases inside the cell. Okay. So this, if it's being turned on, it's going to produce ZAMP. And then it's activation of other kinases. We can also, the effector can also be a phospholipase C. So phospholipase C, which is also called PLC. C, and it's PLC. And when you activate that, it cuts membrane lipids to create second messengers like IP3 and DAG. Okay. So you're actually gonna get IP3 and DAG. Okay. And so those effectors are being activated, and they're gonna do different things depending on what type of effector we have. And then finally, what do we think happens? So we know that the signal, it has to end at some point. And the alpha subunit, it has an automatic timer built into its biological function. It is, obviously, it is an enzyme, right? The alpha subunit is also a GTPase. It's gonna slowly break down its own GTP into GDP. So once the GDP is cleaved back into GDP, the alpha subunit immediately turns off, and it lets go of the effector protein, slides back along the membrane, and reconnects with the beta gamma dimer. And this entire system moves back into step 1. So let me just show what happened. We have this G alpha GTP over here. And now this thing is now being hydrolyzed into GTP. Okay. So we have this one being a GDP. So this is a GDP now. And it's going to detach from here. Okay. We still have this effector over here, and this is gonna detach. Okay. And it's actually gonna slide along the membrane again. And this G beta gamma is also going to slide along. And it's gonna get back to its original place. Okay. So we have this part here. It's gonna slide, and it's gonna go back to its where it is at the start. And so that is that. And this ligand over here too, like once it binds, after some point, it's also going to dissociate. So this ligand is actually going to dissociate. And we are back into step 1. So this is the G protein coupled receptor, or the pathway of the heterotrimeric G protein. That's it for this video. Thanks for watching.